Good day everyone, my name is Lee ZHI XUAN. So this video will explain about the STPM Biology Semester 1 Chapter 2 Organelles topic. So this video will explain about the structure of the lysosome and also its function. So this is the general structure of the lysosome you can see in the diagram. So normally lysosome is the small particle set structure. They containing the hydrolytic enzyme like protease and lipase. And as you guys know that from the previous video, I already explained that lysosome is formed from the Golgi apparatus. So actually it is the part of the Golgi apparatus too. Thus that the cisterna of the Golgi apparatus will be part of to form the unique organelles we call as lysosome. So it containing the hydrolytic enzyme like protease and also lipase. And then the lysosome of the content is acidic, because it based in acidic condition. And normally your hydrolytic enzyme is made from the RER. Of course, it's made from RER. Yeah, why? Because enzyme is protein, right? So all the protein can be formed from the RER and then transport to the Golgi apparatus for processing. And then the primary lysosomes are formed by budding from Golgi apparatus. This is what I just explained lah. Okay, so normally lysosome is more focused on about its function. Because the structure of lysosome is very simple. It's just a circular shaped spherical set structure that containing hydrolytic enzyme. But its function, there are many functions. So one of the function is that it can protect our cell. Meaning that they can digest any nutrient or also waste product in form of food vacuole and phagocytic vacuole respectively through endocytosis. Meaning that when the cells food vacuole is endosized into the cell. So this is all the nutrients. So this is all the nutrients, so that lysosome was starting to fill with this vacuole. This is the food vacuole. The cell normally, one of the examples is the amoeba and the so they were starting to take the food, and once the food is already entered into the cell, the lysosome will fill with the food vacuole to form secondary lysosome. So this one is primary lysosome. Once it fills together, they will form secondary lysosome. And then it will release the enzyme, the hydrolytic enzyme, to hydrolyze all the food particles. So this process is we call as phagocytosis. So not necessarily is food particle in human, like macrophage, the one type of white blood cell, leukocyte. They will undergo a phagocytosis, but this one is not to take a nutrient. This one is to play the role in defense system. They will take up the microbe, the pathogen, the harm, like the bacteria or virus. Then this lysosome will fuse together to break down to kill the bacteria. So this one is if you want to apply in human. But this one is to apply in the paramecium or amoeba only. If you want to apply in human, this one is not food vacuole. It will become the antigen, the microbe antigen. And not only that, lysosome also play the role in autophagy. So what is mean? Autophagy is that when your cells containing the unwanted structure, meaning that your organelles is already old. This organelles is already not functioning, already cannot function, but still remain inside the cell. This organelle cannot remove. They cannot automatically remove one. Like human, right? When human die, we still remain maya, right? We still remain maya. We cannot. If you not do anything, you will not go to the certain place to do anything. Your maya is still there, right? So you need to do anything. Similar to the cells. And the cell, when the organelles is already died, already cannot function, it still remain inside the cytoplasm, on the cytoplasm. So how to remove it? Lysosome play the role of autophagy, which is the process to remove all the unwanted organelles as well. So when the organelles is already unuseful, the organelles were enclosed by single membrane to form autophagosome that later will fuse with lysosome to form autophagic vacuole like this. This one is already mitochondria and vesicle. Mitochondria and peristome is already unwanted organelle. This one is already we cannot use. This organelle is already damaged or they already died, already not functioning. So they will fuse together, fuse to form the one vesicle to form what is we call as autophagosome. This structure is we call as autophagosome, and once the lysosome is fused with this autophagosome, it will form autophagic vacuole like this. And then once it already formed autophagic, as similar in the phagocytosis process, lysosome will release the hydrolytic enzyme to kill the organelles. So the organelles are digested into the molecule, and this digested molecule can be reused to form the new organelles. Because it's still the organelles, still mitochondria. So that although it is already digested, so this digested component still can be used to form the new organelles one. So this process is called as autophagy. Another process is called as autolysis. Autolysis is that when your cell is died, totally died, and our cells have their own lifespan. Not only organelles, cell itself also have its lifespan. So different types of cells have different lifespan. Like erythrocyte, red blood cell have the 3-month lifespan. Only have 120 days only. 120, 4 months. Sorry, 4 months. 4 months. Okay, so whatever type of cell have their its own lifespan. So once when it go to the dead state, there die, the cell also will die. So the cell die, but it still remain a day. If you cell die, but it still remain a day. So how to remove it? Lysosome will take place. So it will release, take place to become the cell digestion. So that cell that containing lysosome, they still containing lysosome, they were starting to release all the enzyme to make the cell die, directly die. So this one is we call as autolysis. So normally, if we want to talk about the function, the lysosome in the human normally is play the role in defense system. So phagocytic vacuole formed by the leukocyte where engulf bacteria will release the bacterial vacuole into the enzyme will be released and to kill the bacteria. Right. This one is where you got about the okay, so that's all for this video. So hopefully you guys can understand about the structure of lysosome and function. So thank you for watching. Bye.